everyone, and thank you for attending today's webinar titled PentaMice, a unique approach to achieve maximum plasma titers for antibody discovery. My name is Matt Harrington, Senior Manager of Operations at Lake Pharma and today's moderator. I am thrilled to host today's webinar. Before we get started, please feel free to submit any questions to our speaker at any time by clicking on the Q&A button located on the toolbar at the bottom of the window. If we cannot get to your question today, we will respond via email. And now I would like to introduce today's speaker to you. Dr. Brian Zabel received his Bachelor of Science degrees in Biology and Mathematics from the Massachusetts Institute of Technology in 1997 and a Ph.D. in Immunology from Stanford University in 2004. Dr. Zabel holds 9 U.S. patents and has published more than 50 peer-reviewed research articles in high-impact journals, such as the Journal of Experimental Medicine. Dr. Zabel has special expertise in hybridoma-based monoclonal antibody discovery with demonstrated success against challenging targets. At Lake Pharma, Dr. Zabel leads a scientific team of hybridoma experts in the discovery of therapeutic and diagnostic antibodies. And now, we turn over to Dr. Zabel.
Thanks, Matt. So, are you interested in antibody discovery? I think you should start with hybridoma.com. This is a website that we created last year in the fall and rolled out. It basically contains 75% educational content on hybridoma-based antibody discovery and about 25% of Lake Pharma's antibody discovery workflow that we use here in the DI, Discovery Immunology business unit. It talks about our campaign successes and our technological innovations. So Lake Pharma is a CRDMO, which is a contract research and development and manufacturing organization. In our group in Discovery Immunology, we work really on the end here, the feather of the arrow that's leading to success here in terms of antibody lead discovery, screening, and lead characterization. So we do all of the hybridoma-based antibody discovery at Lake. We primarily focus on mouse and rat immunizations, and our ALAC accreditation is in progress, which is anticipated next month, May 2020. So just a bit about our business unit and our campaign stats. So the Discovery Immunology business unit is 4 years old. We're focused on discovering therapeutic and diagnostic antibodies. We worked with over 45 clients on an array of challenging targets, from G protein coupled receptors to small molecules, and many of the clients come back for a second, third, or 6 or 7 campaigns with us. We've had a lot of success. We've discovered antibodies for over 100 targets with a high success rate. We've cryopreserved over 10,000 lead confirmed hybridomas, and we have strong capacity to deliver monoclonal antibody diversity. So we've, throughout all of these campaigns, we've sequenced over 700 monoclonal antibodies that have unique heavy and light chain V or J gene usage and or CDR3 sequences. In terms of the targets that we work on, most of them are immuno-oncology related, and then the other bulk of the targets we work on over here are plasma biomarkers, but we certainly have worked on a variety of different things, from neuro to bone to fertility and inflammatory targets as well. So today we're talking about innovations in discovery immunology at Lake, and really, it's all about the PentaMice. So PentaMice are a proprietary royalty-free immunization platform. We use 5 unique mouse strains that we've generated through in-house breeding that cover 9 different MHC haplotypes, and we use 10 mice per immunization cohorts to deliver maximum titers. So here are the stars of the show, our 5 strains of PentaMice. We've got B by S, D by U, K by G7, Q by V, and mixed. And we name them based on their MHC haplotype, specifically related to class 2. So the observations behind PentaMice, over the course of almost 100 campaigns, we've noticed a couple of interesting things. And one of the things that we noticed was in campaigns where we use wild-type mice, we usually use 2 or 3 different strains of wild-type mice. And in an unpredictable way, certain targets would generate high antibody titers in certain strains and would have very low titers in others. So in this particular example, mice from strain number 1 gave us a really strong antibody titer. And what we're looking at here is diluting the plasma out, starting at like 1 to 200 and then going out to 1 to 200,000. And this is an ELISA RLU readout. Plasma from mice that were immunized the very same way, just a different strain, strain number 2, really had a poor antibody titer response. So we know a couple of things. We know that plasma titers are predictive of antibody discovery success. And what we've learned is that there's a strong strain-dependent difference in plasma titers for most targets that's a bit unpredictable. So the concept behind PentaMice is that it's known that high plasma titers require T-cell help. So to get a good antibody response in vivo, where we have affinity maturation in germinal centers that are happening in vivo, that requires really robust T-cell help. And that's actually where the CD4 helper T cells get their name, is they provide this help to help the B cells basically develop and expand in vivo and secrete high affinity antibodies. A key component of being able to deliver that T cell help is the requirement for recognition of cognate antigens presented by MHC class II. So the class II receptor is expressed on B cells, and the T cells, the TCR recognizes peptides in the context of class II. So MHC class II is highly polymorphic and possesses different peptide binding and presentation profiles. So we hypothesized that MHC polymorphism is the underlying mechanism that's driving the strain-dependent differences in plasma titers that we're seeing over the course of 75 campaigns that we've run. So in this example, we have a peptide 1 here, and these are our 5 different PentaMice strains. Peptide 1 is sort of indiscriminate. It can be equally presented by MHC from any strain of mouse, basically, any of the 5 PentaMice strains. In contrast, peptide number 2 here is only effectively loaded and presented by the K by G7 strain. And so if you didn't have this strain or this sort of MHC molecule in your wild-type mouse repertoire, you wouldn't get a good antibody response in vivo. So, which led us to the PentaMice. And so the question is, how can we maximize immunological diversity while maintaining practical group sizes? Because it would be great if we could just order up all of the different strains of wild-type mice that JAX has and run a 100 mouse campaign, but really 10 mice per immunization cohort is in the Goldilocks zone in terms of requiring a practical amount of immunogen needed to immunize the animals to supply enough B cells to support multiple fusions. So our approach to basically maximize MHC diversity is to cross inbred strains of different MHC haplotypes and then use the resulting F1 offspring. So in this example here, you've got a syngeneic animal that expresses, MHC haplotype of the B, and this is the K, and the offspring express both haplotypes, so B and K. So it's just standard mother to father, genetic inheritance for the MHC class II genes. In addition, this is a pretty straightforward approach that can generate an immunization cohort with increased MHC class II diversity. So we got 5 strains total, 2 mice per strain. So in addition to that, we selected certain autoimmune prone strains to have sort of a built-in tolerance breaking into the mice that we use. In terms of MHC class II molecule, you see in the diagrams here, it's a heterodimer. It's actually an alpha and beta chain. So MHC class II in mice, there are 2 different gene loci that encode class II. It's the IA and IE loci. So each of these encode an alpha and beta chain. So in a conventional, regular old C57 black 6 mouse, actually an IE null, so it only expresses the IA class II genes of the B haplotype. So there is one sort of class II molecule, heterodimer, that's expressed by a B6 mouse. Now, our PentaMice strain, we do have that haplotype as well, but we also have 41 additional MHC class II molecules that can be expressed as part of the PentaMice repertoire. So this represents a 42-fold increased MHC class II diversity over if you were just to use just a regular old B6 animal. So we did some immunophenotyping just to characterize our PentaMice. And one of the first things we did was look at the B cells by CD19 and then look at CD45 allotypes. So all leukocytes express CD45, and there are two different allotypes that they can express either CD45.1 or CD45.2. So if you look at the B strain here, these guys express CD45.1. The S strain expresses CD45. This one expresses CD45.2. The S expresses CD45.1. And if you look at the B by S, these guys are expressing both CD45.1 and 0.2. So clearly these are the F1 offspring there. And similarly for the other strains as well. I think actually the D by U both express CD45.2, so their offspring is only CD45.2. So we did some additional work to look at the MHC class II immunophenotype. So there are 4 commonly available antibodies that recognize different alleles of the MHC2 gene. And they're listed here by the antibody clone, so IAIE, IAB, IAD, and IAKA. And depending on your mouse strain, you'll express different binding profiles, basically, for these 4 antibodies. So we use these antibodies to immunophenotype the B cells. For example, looking at the K strain, so this is one of the parents, it expresses the first one here and the last one. So you can see it expresses IAIE and IAKA. The G7 only expresses the IAD. You cross them together, and now the resulting offspring express the first column, the third, and the fourth sets of MHC molecules. So we actually know quite a bit about the PentaMice, and we've characterized them pretty in-depth in terms of MHC. We also know a bit about the antibody secretion. So in terms of IgG2 subclass characterization, this is kind of interesting. Most evidence indicates that mouse IgG2A and 2C are mutually exclusive in plasma. And there's a paper by Zhang et al. That proposes that these 2 subclasses are actually allelic. And I think our data from the PentaMice indicates and supports the allelic idea. So if you look in the plasma from the Q animal, for example, you can find lots of IgG2A. From the V animal, you see IgG2C, and in the cross, you can see both IgG2A and 2C. So let's look at PentaMice in action. So Lake Pharma is working hard to be part of the solution for COVID-19. And the company's been generating spike protein for use by the research and medical communities. And we've taken advantage of that in our hybridoma group as well. So given the immunological diversity of the PentaMice platform, I think it's much more representative of outbred humans, for example, than the single strain of inbred mouse that are typically used in immunization or vaccine preclinical studies. So we applied our PentaMice platform in an immunization approach. We also added in 2 BALB/c animals for comparison. And this is what the day 9 titer looks like using Lake Pharma's expressed SARS-CoV-2 spike protein. And you can see here there's an impressive early day 9 IgG titer. And what we see right away is there's also some strain differences. So the B by S strains in gray are consistent and giving a superior response. And it's actually quite different than the early response given by D by U in purple, for example. So I think that's interesting. The BALB/c are sort of middle of the road here in terms of an IgG response. And overall, I think the PentaMice can provide a unique and immunologically diverse preclinical platform to study COVID-19 vaccine responses, certainly more representative than a single inbred strain would be. And just in comparison here to rabbit polyclonals that are available commercially, using 10 microgram per mil, the signal here is only about 100,000 RLU, and the polyclonal sera that we're getting from our animals all really exceed that. So we're getting very strong titers in these animals. So the next step in the COVID-19 project here is we're looking for inhibition of human ACE2 binding to the SARS-CoV-2 spike protein by plasma from our immunized or vaccinated PentaMice, if you will. So the first step in viral entry is the spike protein, which is in red here, binding to the human receptor angiotensin converting enzyme 2, which is here. And so we've devised an ELISA biofunction assay to recapitulate this in vitro. So you have an ELISA well, we have our human ACE2 that's produced here at Lake with the human FC tag. We come in with the spike protein that has a His tag, and then use an anti-His HRP antibody to detect binding. So in our animals, if we have generated antibodies that can bind to the spike protein and inhibit the binding here, and we predict to see a lower signal, basically a lower anti-His signal. And that's what we see over here. So if we use control plasma and take that as the max signal in the assay, you can see that plasma at a 1 to 200 dilution from our Q by V, B by S, and B by S strains is able to really substantially inhibit the binding there of the spike protein to the humanase 2. And these neutralizing antibodies are generated as early as day 9 here in this study. So we're hoping, our work here to really be part of the solution here and add to the research and medical communities, everybody working on SARS-CoV-2. So some other examples of PentaMice in action. So these are 4 different targets that we had immunized animals with. The strains are here and they're grouped by color. And you can see in this particular campaign, the D by U strains and the B by S strain, and the K by G7 strains in blue, they didn't do very well, but in comparison, the mixed strain in green did okay, and boy, the Q by V looked really good here, so giving a very strong titer response. In this campaign, the top responding animals are B by S and our D by S, in gray. Again, Q by V is sort of middle of the road, and in these animals you do see a better response though from the D by U. So again, it's kind of unpredictable depending on the target which strain is going to be the winner. For this particular campaign, nearly all the animals seem to respond well to the immunization protocol that we use. And in this particular one, it looks like the mixed strain didn't do as well. But the other strains look pretty similar. Maybe the D by U didn't do as well either. So from each of these campaigns, we discovered 75 to 375 lead hybridomas. And I think what we were finding out is that they've yielded a diverse array of target-specific antibodies. So in summary here, the PentaMice platform is a proprietary, royalty-free immunization platform designed to deliver maximum plasma titers. In terms of immunological diversity, if you compare it to a B6 animal, we express 40 times as many different types of MHC class II heterodimers. In total, there are 5 unique strains that we generate through in-house breeding. 9 MHC haplotypes are covered, 10 mice per immunization cohort. And I think it's a defined immunologically diverse preclinical platform to study COVID-19 vaccine responses. So if you're interested in PentaMice or hybridoma-based antibody discovery, please shoot me an email, give me a call. More information is available on our website. And since there's a couple extra minutes just for fun, I thought I would point out how we came up with the PentaMice name here. So in terms of branding, PentaMice versus Muse mice versus the Great Lakes platform. So we landed on PentaMice. We have the strains here. It seems a little bit more, scientifically justified by using the haplotype initials. But another idea that we liked was the Muse mice, because the mice are our muse, our inspiration, and the Greek muses were also known as mouses, and we had 5 strain names for these guys as well. And then we have the Great Lakes platform, GLP platform, because Lake Pharma. And we have the strain names here. And also it seems like the Great Lakes have the shape of mice or rodents, at least if you use your imagination there. So that is where I guess we would like to end. And in terms of acknowledgements, I'd like to thank the Discovery Immunology Group, Ashley and Meow. They did the PentaMice fax and ELISA characterization. Xiaomei and Dan, who've been working on the COVID-19 R&D project. Natalie Giuliani is our vivarium specialist and does all the PentaMice breeding. Sophie, Matt, Guillaume, Ling, and Joyce are scientists in the Discovery Immunology Group running all of our campaign work. Margaret Wong, who is currently now head of antibody engineering at Lake. Carrie, Ala, Ankush, and Baidehi, who are our scientific managers, and Kavya, Jessica, and Lily, who have helped out with marketing. So again, if you're interested, please contact me, Brian Zabel at lakepharma.com, and go to our website for more information. And we can open it up for questions now. Yeah.
Thank you, Dr. Zabel, for your presentation. For those who joined late, we will also be making a recording of this presentation available online. Now, as Dr. Zabel mentioned, we would like to open up the floor to questions. As a quick reminder, you can submit questions by clicking the Q&A button located on the toolbar at the bottom of the window. If we don't get to your question during the broadcast, we will follow up with you offline. So our first question that came in for Brian, how often do you see such a marked difference in serum titers as you've seen in the earlier slide from strain 1 and strain 2? And do you have a percentage from the AG you have done so far?
I would say maybe 10% of the campaigns we see a really dramatic difference in the plasma titers like we saw in that first slide. I showed another example earlier, or in the, I think it was one of these other slides, where the D by U and the other strains didn't perform as well. So it can happen. Anyway, I don't have to go back to it, but, and what was the other part of the question, Matt? Oh yeah, here it is.
Do you have a percentage from the AG you've done so far?
From the age?
AG. Ag.
From the antigen that we've done so far. You mean in terms of different antigens? So I think we've run around 15 different PentaMice experiments, with the full cohort of PentaMice. And of those, I think most of them look like this here, and the other 5 will be some version of this or this.
All right, our next question that came in, did any particular strain result in greater sequence diversity of final clones?
No, we didn't see that. We see really good diversity independent of the strain that we used so far.
Does the PentaMice platform produce humanized MABs directly?
No, it does not. This is completely wild type, and that's one of the reasons it can be royalty free. Is these are just animals that we breed in house. There's no fancy transgenics involved. Lake Pharma Antibody Engineering can humanize any of the wild type antibodies that we discover, and that's actually a pretty common approach for therapeutics. Therapeutic discovery. So if you go that direction, then you're not tied into the milestones and royalties of using the transgenic human antibody producing mouse strains. So it's a pretty good option. It really depends on it's a business decision, I would say.
By day 9, do you see a diverse repertoire and high affinity?
So day 9, that's probably referring to the SARS-CoV-2 antibody. And affinity is something that you really need purified antibody in order to put a number on that. So we can't really talk. We can talk in general about affinity, but we can't put a KD number on that because we haven't purified the antibodies from the polyclonal soup. But yeah, certainly we're getting good binders and good neutralizers even at day 9, which is a very early time point. So my feeling is that the spike protein there is very immunogenic. It's on the end of, on the spectrum of titer responses, it's very antigenic in comparison to some of the other projects that we've worked on.
Could you comment on any affinity or diversity differences between the strains?
No, not really. I think we end up pooling material from the strains in most cases, and when we do that, if we pool lymphocytes, for example, from 2 or 3 strains, we can't track back and say this came from this strain or this antibody came from this strain. I think our thinking about it is what sort of what combination of animals or material from the animals can give us the best sort of antibodies. And moving forward into the hybridome and the fusion, we don't really track it at that point in terms of the strains. What we do track sometimes is, let's say we immunize a group with DNA versus a group that's immunized with protein. We'll keep those separate, and but within those 2 different immunization cohorts, there'll be a combination of different PentaMice, material from the PentaMice. But then you can go back and track back and say, okay, so these high affinity antibodies came from the DNA immunization group, and this other group of really good functional blockers, they came from the protein immunization group. So that's a sort of granularity that we can get to. It would be great if we had a project where we could look at the individual, PentaMouse diversity and affinity from each of the strains, like head to head. That would be a fun R&D project, but it's not something that we have any data on currently.
Could you elaborate a little on the size difference in amino acid residues between peptide fragments presented on MHC molecules and the binding site epitopes of antibodies? Is there any correlation?
Let's see. So for MHC class 2, the peptide binding size is, I think, between 13 to 18 amino acids, and then in terms of the antigen binding, the epitope binding size for an antibody, I've heard it's like 6 to 11 amino acids. I don't know that they correlate very well, because we've made antibodies against haptens, which are much smaller than, or they can be smaller than amino acids. So I don't think there's a correlation there, but yeah, that's a little bit of an answer for that question anyway.
Now that so many transgenic mice are available, why would wild type be the way to go?
Yeah, I think kind of answered that before with respect to the milestones and the royalties that are involved in using the human antibody producing transgenic lines. I think those strains of mice, they work really well in our hands, and so it can be a fast approach because you don't have to do the humanization. But then, it's a business decision, because you might now be giving up some X% of, any of the royalties that you would otherwise be getting if you used wild type mice. So wild type mice is, and PentaMice in particular, for us, they're royalty free. And so the next step then basically is humanization. And although there is some cost and time involved in humanization, there are no milestones or royalties involved in paying out then to use your humanized antibodies down the road. So that would be one advantage of going that route of using wild type mice. In other cases, sometimes people really need a preclinical tool antibody that you would want to use as proof of concept in vivo. And so using wild type mice or rats to generate those reagents, there's really no need to have a humanized antibody for those types of projects. And finally, if you're making diagnostic antibodies, you really just need wild type antibodies. I don't think there's a great need to have a humanized or a human version of an antibody for diagnostics.
There was a recent article entitled Clinical Trials Will Have to Evolve with Our Evolving Population, where the author brings up the importance of MHC diversity and the clinical trial patient pool. Were you inspired by such literature in the creation of PentaMice, and can you comment about the importance of MHC diversity on both the preclinical and clinical fronts of drug development?
Yeah, I think we were inspired mainly by what we were seeing in our, in our campaign work, where we used different wild type strains and just saw different immune responses. But that's certainly, when we talk about sort of preclinical vaccine development, let's say around COVID-19 projects, I think you'd be in much better shape looking for immune responses in the PentaMouse strain, which at least makes an attempt to model the diversity you would see in outbred human patients. I think for humans, MHC diversity is very large, in the tens of thousands. If you look at all of the inbred wild type strains of mice, probably the maximum allelic diversity is like 200. So with the PentaMice, we're capturing a good portion of that, and we're at least making an attempt. And I think in our first sort of immunization and vaccine slash vaccination attempt with the spike protein, we see strain-dependent differences early on at day 9. So it does show that I think the immunological diversity, which we're thinking is MHC dependent, really can drive your responses. So yeah, I think it's a strong platform that would behoove people to consider when looking at vaccine development.
All right, we'll go to one final question because we're about at the time limit here. Are you proposing using all 5 strains for every program, or are you able to predict which ones to use?
Yeah, we would do all 5 strains for every program. So it's 5 strains, 10 mice. And we'll do that for a protein cohort. Let's say we want to do protein and DNA, so that would be 20 mice total. And it really funnels down into just picking the best mice with the best titer. So I think it just gives you more opportunities for chances of success. So I don't know that we can predict, we know something about GPCR targets, but I don't think we can predict a particular strain will always work well with a particular class of target. So I would vote to do the whole, the PentaMice platform for each incoming target per each immunization method.
Excellent. Thank you, Dr. Zabel, for your presentation, and thank you to all of you in the audience for joining us this morning. This concludes today's presentation, and again, a recording will be made available online shortly.